So we are officially back after a short break, and welcome to the next episode of the Worm Podcast.
It is really good to be back.
Yeah, it really is. And before we jump in, just a quick reminder to all of you listening, please make sure to follow, like, comment, and share this with people to help grow the community. We really appreciate it.
It makes a huge difference, yeah.
It totally does. Also, if you are doing some fascinating work in this space and you'd like your research featured on a future deep dive, please reach out and contact Dr. Viren Chauhan. All of his details are right down there in the show notes.
Definitely reach out.
Okay, so right now, as we speak, scientists around the world are trying to build this ultimate map, like a map of every single protein in a living animal.
Right.
But the wild part is, at their current pace, it's going to take at least 100 years to finish.
Which is, not ideal.
Not ideal at all. So today, our mission for this deep dive is to look at a biological cheat code that basically condenses that entire century of work into just a few years. We are exploring a blueprint for creating a genome-wide visual atlas of an entire living organism.
And since y'all are already pretty familiar with our favorite microscopic worm, C. elegans, we can skip the basics and just dive right into the really good stuff.
Exactly. We're pulling from this fascinating pilot study by researchers Matthew Aroglu and Oliver Hobart, along with an eLife assessment. And they detail this revolutionary strategy for whole proteome tagging.
It's a massive leap forward.
It really is. So, okay, let's unpack this. We already mapped genes, right? Like, we have the genome.
Sure, yeah.
But genes are just, they're just an instruction manual. Proteins are the actual physical machines doing the work inside the cell. So to truly understand biology, we have to actually watch the machines work.
Right. To set the stage for you here, imagine the backdrop behind us is this high-tech microscopy lab.
Okay, I see it.
But as we get into the details, the walls kind of just fade away, and they transition into this glowing, color-coded, three-dimensional map of a microscopic C. elegans worm.
Oh, wow.
That glowing map is the ultimate objective here, because the fundamental problem driving this whole research is that transcript data, the messenger RNA that tells us a gene has been activated, it just does not perfectly match the actual proteins present in a cell.
Wait, really? It doesn't match?
Not perfectly, no. I mean, the mRNA is basically just the cell's intended order form.
Okay.
And a lot of biological bureaucracy happens between placing that order and the final protein actually being built and deployed.
So the RNA says, hey, build this, but it might not actually happen.
Exactly. So to really understand what a cell is doing, we have to look past those order forms and actually watch the proteins in action inside the living tissue.
Which brings us to the absolute logistical nightmare of actually building this glowing map.
It's done.
Yeah. So currently, out of the roughly 20,000 protein-coding genes in C. elegans, only about 1,554 have been endogenously tagged.
Yeah, that is a tiny fraction.
It's literally just 8% of the entire proteome. And before we go further, let's just clarify that term for a second.
Sure.
When we say endogenously tagged, we mean altering the worm's own native DNA and its chromosomes, right? We're not just injecting those floating temporary rings of DNA.
Right. That is a really crucial distinction. We are talking about permanently editing the organism's native genome.
Okay.
So that every single time it naturally builds a specific protein, it automatically attaches a fluorescent tracker to it.
Like a little glowing beacon.
Exactly. But doing this one by one is just agonizingly slow.
Yeah.
And the systemic issue here is that the labs doing this work are, they're completely uncoordinated.
Yeah, there's no central boss telling them what to do.
Right. There is no centralized push. Individual researchers are just tagging the specific proteins that they happen to need for their own isolated experiments.
Which makes sense for them, but for the big picture, it's terrible.
It is. The sources actually note that highly studied genes, like PGL1 or DAF16, they have been independently tagged at least 4 different times by entirely different labs.
Oh, man. So it feels like trying to build this massive, intricate mosaic, right?
Yeah.
But all the artists are just blindly painting their own favorite tiles over and over again. And they're leaving the rest of the wall completely blank. It is just incredibly inefficient.
It really is.
But Aroglu and Hobart's proposed solution is what completely changes the math here. They are utilizing pooled CRISPR-Cas9 injections.
This is where it gets really smart.
Yeah, so instead of carefully injecting a single gene tag per worm, they are simultaneously tagging 3 distinct genetic loci all at once.
3 at a time.
But I actually have to push back a little here. Like, if CRISPR technology is so advanced now, why stop at 3?
That's a fair question.
Right. If we want to map 20,000 genes, why not inject a pool of, I don't know, 20 tags at once and just finish the whole mosaic this year?
Well, the limitation isn't our ambition. It is the biological tolerance of the organism itself.
Okay.
We have to remember how CRISPR actually works in practice. Cas9 is basically molecular scissors.
Right, it cuts the DNA.
Exactly, cuts the DNA at a specific site. But CRISPR relies entirely on the cell's own internal repair machinery to stitch the DNA back together.
Oh, so the cell has to do the heavy lifting.
Yeah, the cell has to incorporate the new fluorescent tag that we introduced during that repair process. And these fluorescent tags are massive pieces of genetic code.
So big.
Very big. So if you introduce 20 cuts across the genome all at once, and you flood the cell with 20 massive repair templates, you completely overwhelm its homology-directed repair machinery.
Like giving it way too much homework.
Exactly. The biological tool is just too high. The cell will either completely fail to incorporate the edits, or the trauma will simply kill the worm.
Ah, okay. So 3 is basically the biological sweet spot before the worm's internal repair system just throws its hands up and quits.
Precisely. And if we connect this to the bigger picture here, a factor of 3 is actually a massive mathematical leap.
I guess so, yeah.
Going from 1 to 3 sounds incremental, just 2 more. But when your finish line is 20,000 genes, reducing the workload by 2/3, it takes a century-long fragmented slog, and it condenses it down into a single generation of scientific effort.
That is wild. It takes the impossible and just makes it a scheduling problem.
Basically, yeah.
But that raises a massive technical hurdle, right? How do you actually sort through the results?
Right.
If you inject 3 different tags into a microscopic worm, how do you know which ones actually worked? Because normally, researchers rely on something called a co-CRISPR marker, right? It's essentially a visible flag, like intentionally giving the worm a mutation that makes it twitch, just to prove the injection needle successfully delivered the payload.
But here's the problem with that. If you add a twitcher mutation, or really any extra co-CRISPR marker, on top of 3 massive fluorescent tags, you are overloading the cell's repair system all over again.
Oh, right, because that's a 4th edit.
Exactly. So Iroglu and Hobart had to figure out how to see their edits without using those traditional visible flags.
So what do?
Their solution is this highly rational, color-coded strategy.
I love this part.
It relies on matching the predicted expression level of a gene with the inherent brightness of 3 specific, very modern fluorescent fluorophores.
Okay, and they measure that predicted expression using a unit called FPKM, right?
Yes, FPKM.
Let's break that down a bit because it sounds like some heavy jargon. So FDKM stands for fragments per kilobase of transcript per million mapped reads.
It's a mouthful.
It really is. But basically, it's just a metric for counting RNA transcripts. So it tells researchers, hey, this cell is pumping out a massive amount of RNA for this specific gene, so we expect a massive amount of protein to be built.
That is the perfect way to look at it. It is a volume metric.
Okay, volume.
And using that volume metric, they assign their colors. So they use a blue tag, specifically M tag BFP2, exclusively for highly expressed genes.
Okay, so the really busy ones get blue.
Right, those with an FKKM over 500. Then they use a green tag, MST-Gold, for modestly expressed genes sitting between 100 and 500 FPKM.
Got it. Blue is high, green is medium.
And finally, they use a red tag, M-Scarlet3, for the absolute lowest expressed genes, anything with an FPKM of 18 to 100.
Okay, but wait, why not just make them all the brightest color? Like, why assign the colors based on volume?
It's all about the signal-to-noise ratio.
How do you mean?
You see, C. elegans worms, they naturally emit their own background light under a microscope.
Oh, right, autofluorescence.
Exactly. And the OP50 bacteria that they eat, that also emits background light. Even the agar plates they live on glow slightly.
That's annoying for scientists.
Very annoying. And almost all of this natural biological background noise exists in the blue and green visual spectrums. There is very little natural red autofluorescence in these worms. Therefore, the dimmest genes, the ones producing a very tiny number of actual protein molecules, they need the absolute highest contrast tag to be seen against that background.
So they have to be red.
They have to be red.
Here's where it gets really interesting, right? Think of it like trying to spot different light bulbs in a massive glowing city at night.
I like this.
You can use a blue light bulb on a stadium-sized billboard because there are thousands of bulbs packed together. The sheer volume of that blue light is bright enough to cut through the ambient glare of the city.
Yes.
That is your highly expressed gene. But if you are searching for a single firefly, like a low expression gene, it better be glowing bright red. Because if that single firefly glows blue or green, it will get completely lost in the ambient streetlights.
I love that analogy. It perfectly captures the physical reality of looking through these microscopes.
It makes so much sense.
And the elegance of this design is how it completely streamlines the workflow. By tying the tag color to the natural protein volume, researchers can screen the results incredibly fast.
How fast?
Well, they just look at the first generation of worms, the F1s, under a standard low-magnification fluorescence stereo microscope.
Oh, so they don't need the crazy high-powered stuff yet.
No, because the bright blue tag acts as an anchor. If they see bright blue cells, they know the CRISPR injection was successful for that specific pool.
Oh, that's so smart. The blue just proves it worked.
Exactly. And once they have that visual confirmation, then they can zoom in with higher power to look for the dimmer green or those tiny red fireflies. They completely eliminate the need for those disruptive twitcher markers.
It is an incredibly clever design, so the logic is totally sound on paper.
On paper, yes.
But biology notoriously hates following human logic. So when they actually bred these edited worms in the pilot study, did the glowing proteins show up where the RNA predicted they would?
The short answer is a resounding no.
Wow.
Which is exactly why building this visual atlas is so urgently needed. In the pilot, the researchers attempted to tag 30 specific genetic loci, divided across 10 pools of 3. They successfully isolated 24 of those 30 tags. And when they finally observed where these proteins were actually living and working inside the worms, they found massive discordance between what the single-cell RNA sequencing had predicted and biological reality.
The tissue surprises alone are wild in this paper.
They really are.
Let's look at the protein HXK1. It is a glucokinase, basically an enzyme involved in basic sugar metabolism.
Right.
Based on the RNA data, you would expect this blueprint to be everywhere, maybe heavily enriched in high-energy tissues like muscle or neurons. But when they looked at the glowing red proteins, it was actually highly enriched in the gonadal sheath.
It's totally unexpected. And we see a similar mismatch with ACDH10, which is an enzyme for fatty acid metabolism.
What happened with that one?
The RNA sequencing predicted it would be localized in the intestine and epidermis. But the actual protein map showed it was broadly distributed across the worm's somatic cells, yet heavily depleted in the germline.
But how does that actually happen physically? Like, how can a cell have a massive stack of mRNA blueprints for an enzyme, but when you look for the final protein, it's just not there?
It comes down to active post-transcriptional regulation.
Okay, what does that mean in practice?
Well, the mRNA is just a blueprint sitting on the cellular factory floor. But depending on what that specific tissue needs at that exact moment, the cell might use microRNAs or specific ubiquitin degradation pathways to actively shred that blueprint before the protein is ever built.
So they just rip up the instructions?
Basically. Or it builds the protein and immediately degrades it. The RNA data says the factory intends to build this, but the protein tag tells us the factory manager stepped in and canceled the order.
That makes total sense. It's the difference between intent and execution.
Exactly.
But the surprises weren't just about which tissues had the proteins. The subcellular surprises, like where the proteins localize inside a single cell, that fundamentally shifts how we understand cellular architecture.
This is probably my favorite part of the study.
Yeah, the study highlights two proteins, GDH1 and CARS1, and both of these were firmly established in the literature as mitochondrial proteins.
Right. And logically, you would assume that if you tag them both in the exact same worm, they would overlap perfectly, right?
Yeah, they should both just light up the mitochondria together, acting as generic parts for the cell's powerhouses.
But when the researchers looked closely at the gonadal tissue, the two proteins didn't overlap at all.
Not at all.
No, they localized to entirely different subsets of mitochondria within the exact same tissue.
Wait, so mitochondria inside the same tissue aren't just generic batteries. They have specialized, distinct metabolic identities depending on exactly where they physically sit.
Yes. What's fascinating here is that this aligns beautifully with very recent findings regarding metabolic tuning.
Metabolic tuning.
Yeah, different geographical zones within a cell have different energy and metabolic demands. So the mitochondria are actually differentiating to meet those highly specific local needs.
That is amazing.
And if we had just kept relying on RNA data from, crushed up worms, these specialized subgroups of mitochondria would remain completely invisible to us.
Because the RNA just says, hey, we have mitochondrial parts.
Exactly. The RNA just tells us the battery parts are present, but the protein tags tell us how the cell actually customizes and organizes those parts.
That level of microscopic compartmentalization is just stunning.
It is. It also completely changes how we view paralogous genes.
Paralogs, right. Remind us what those are.
Paralogous genes are duplicate genes that evolved from a single ancestral gene, and they often maintain similar functions.
Okay, like twins doing the same job.
Right. And when you look at RNA data, it often shows multiple paralogs existing broadly across a tissue, seemingly doing the exact same job. But when you tag the actual proteins, you start to see that these duplicates are subdividing the labor.
Oh, interesting.
Yeah, one paralog might handle sugar metabolism strictly in the intestine, while its twin handles the exact same process, but exclusively in the gonadal sheath. We are revealing the hidden, specialized architecture of life here.
So what does this all mean for the roadmap ahead? Because we've seen the brilliant design and the immediate biological payoffs of this pilot study, but we have to address the reality of scaling this up to 20,000 genes.
The scale is definitely the next challenge.
Because as you mentioned earlier, they attempted 30 tags and isolated 24. That is a 20% failure rate.
Right.
What is the physical hurdle we are hitting there?
The primary hurdle is the disruption risk of what we call a translational fusion.
Okay.
The researchers deliberately targeted essential genes for this pilot. That means if attaching the fluorescent tag broke the protein's function, the worm simply died.
Right, a lethal phenotype.
Exactly.
And it makes sense why it breaks, honestly. We are essentially attaching a giant glowing backpack to a microscopic machine.
That's a very good way to picture it.
Yeah, attaching a full fluorescent protein to a native protein can cause what chemists call steric hindrance. Like, think of a protein like a complex folding car engine. If you weld a giant glowing bumper to it, you might physically block the gears from turning or prevent it from connecting to other machines.
Exactly.
Yeah.
And the researchers try to mitigate this by using an 18 amino acid linker.
What's that do?
It's essentially a flexible tow cable between the native protein and the glowing tag.
Ah, to give it some slack.
Right, but clearly, if 20% of them are still failing, the tow cable isn't a silver bullet.
So how do we map that missing 20%? Do we just leave them out?
Well, this raises an important question about our alternatives. One approach the sources discuss is using much smaller epitope tags, like 3xFLAG or ALFA tags.
Small tags, okay.
Yeah, instead of welding a giant glowing bumper to the protein, you are just attaching a tiny molecular sequence, like a small sticker. It is far less likely to cause that steric hindrance.
But there is a massive trade-off there, isn't there? You can't do live imaging with those tiny stickers, right?
Correct. That's the catch. To see a 3xFLAG tag, you have to chemically fix and kill the worm, then wash it with specialized antibodies that bind to the sticker and emit light.
Oh, man. So you completely lose the dynamic, real-time visualization that makes this entire atlas so powerful in the first place.
You do. You lose the live action.
So how do we keep the live visualization but lower the failure rate?
This is where modern computational biology steps in.
Ah, AI.
Yes, exactly. Before researchers even touch a needle, they can use AI tools like AlphaFold to predict the three-dimensional structure of the tagged protein.
Oh, that's smart.
Yeah, proteins have an N-terminus and a C-terminus, which is essentially a front bumper and a rear bumper.
Okay.
If AlphaFold predicts that attaching the fluorescent tag to the front bumper will bury the protein's active site, the researchers can redesign the CRISPR edit to attach it to the rear bumper instead.
So they basically test fit the glowing backpack in a simulation before doing it for real.
Exactly. But even with AI assistance, the authors conclude that directly fusing these large fluorophores, despite the failure rate, remains the absolute best balance of high-throughput speed and direct live visualization.
And the speed is what truly blew my mind in this paper. Let's just look at the math the study lays out for a second.
The math is very encouraging.
Yeah, if a single lab microinjected just 30 worms a day, targeting 18 genes across 6 pools, they could cover the entire C. elegans genome in about 5.6 years.
5.6 years.
Not a century, 5.5 years. And they calculated the material cost at roughly $109 per gene.
Which brings the total material cost for the entire 20,000 gene proteome to roughly $2.2 million.
Right.
Now, one lab spending $2.2 million and 5 years doing nonstop microinjections, That is an incredibly grueling prospect.
Nobody wants to do that, but it is an absolute bargain for the global scientific community.
Absolutely.
If you organize a distributed network of labs, say 20 labs around the world, and everyone is taking a specific chunk of the genome, and they're sharing their successful strains in a centralized database like the WormTag DB mentioned in the paper.
Yes, WormTag DB.
This is highly feasible. It's totally doable.
That is the core message of this research. It isn't just presenting a few interesting new glowing worms. It is issuing a logistical call to action.
Yeah.
It proves that the CRISPR efficiency, the color-coded screening methodology, and the economics are all perfectly aligned right now to build this comprehensive atlas in our current decade.
That is incredible. So, to sum up what we've explored today, transcript data is really just the script, but the proteins are the actual physical performance on stage.
Well said.
By leveraging a highly logical three-color multiplex CRISPR strategy, Oroglu and Hobart have provided the scientific community with a scalable, highly efficient roadmap to light up every single protein in a living organism.
They really have.
They have given us the blueprint to move from just reading the text of life to actually watching it perform in real time.
And I want to leave you with a final concept to mull over.
Lay it on us.
Throughout this deep dive, we have mostly talked about proteins working inside their own cells, like those specialized mitochondria.
Right.
But the sources note that roughly 17% of the C. elegans genome encodes for secreted proteins.
Secreted, meaning things that leave the cell, like hormones and neuropeptides.
Exactly. Now, imagine a near future where this entire tagging system is complete.
Okay.
We wouldn't just be watching where proteins live inside a static, isolated cell. We would be watching a glowing, real-time network of neuropeptides and hormones physically traveling between cells in a living animal.
That is mind-blowing.
We would literally be mapping the physical, real-time communication network of life itself as the organism thinks, moves, and reacts.
Wow. From uncoordinated artists painting identical mosaic tiles to a fully illuminated, moving blueprint of a living creature's internal communication, it is a powerful reminder of just how much of biology is still waiting to be truly seen. Well, thank you so much for joining us on this deep dive, and we will see you next time as we keep exploring.